All right, it's 2 minutes past 11. Let's go ahead and get started. So today's lecture is about pathway and functional enrichment analysis methods. And that's probably one of the most important lecture you will be hearing, because I will be talking about pathway and functional enrichment analysis in terms of interpreting your data. So that's what I will be talking about. Why we care about enrichment analysis? What is it? I will talk about gene ontology and pathway enrichment analysis, and we'll talk about some tools and references. So first of all, why do we care about enrichment analysis? You all know that human genome contains like from 20 to 25,000 genes, and each gene has multiple functions. And if you do your experiment, and if you compare experimental condition, you may identify like a handful of genes. For example, 1,000 genes may have changed their expression. And your question, obviously, what those 1,000 genes do? That's a big question mark. How do we go with that? You probably heard the words birds of a feather flock together. And that corresponds to genes that change together under particular experimental condition. So if you put your yeast in hypoxic condition, genes that may be changing their expression, they may be responsible for hypoxia response. And that's the basis of functional enrichment analysis. You look for genes that change together, and you are looking which functions they are sharing. So you look for common functions that are shared among differentially expressed genes or genes that have been that are changing under any experimental condition. And it's not about genes that have similar genes that with similar expression patterns, they share similar functions. It's also about people. People with similar genetic patterns are also likely to share some similarities. For example, they can be friends. And this boils down towards answering the question, what's so special about your genes that changed under particular condition? Again, we have genes that changed their expression, and based on prior knowledge, that's what we will be focusing about. You make a conclusion what your genes are doing. Using this prior knowledge, those databases I will be covering, you perform enrichment analysis to find out that your set of genes share functions that correspond to coffee addiction pathway. That's pretty much my addiction, so hopefully it's not yours. That's enrichment analysis, how it works. You have your genes, and as I mentioned, each gene has multiple functions. And you look whether some functions are shared among all those genes. For example, you can see that gene 1 has function 1 and 2, gene 2 has function 1 and 3, and the last gene also has function 1 and 5. And you can see that function 1 is actually shared among those genes. That's what you are looking for. You are looking for the functions that are shared among all your genes that have been identified as differentially expressed or under some other experimental conditions. The formal statistical definition of enrichment analysis is the detection whether a group of objects has certain properties more or less frequent that can be expected by chance. What do I mean? If you look at those two jars, one jar and another jar, you can see in the first jar you have similar proportions of red and black balls. So that's what you would expect by chance. In the second jar, you can see that you can see some disbalance of red balls. There are just more of them, and there are few black balls. That's what you can look at it and see. There is some kind of enrichment of red balls in this second jar. Now we need to define some statistical definition for it, and we will come to it. So those jars, consider them like genes that share certain functions. For example, jar 1 may correspond to hypoxia pathway. Jar 2 may correspond to some tumor suppressor pathway. And red balls correspond to genes that are differentially expressed. So in the first jar, in the hypoxia pathway, you don't see very much enrichment of red genes. But in the second jar, which corresponds to tumor suppressor pathway, you can see some enrichment of differentially expressed genes. To do enrichment analysis, we need to define those gene sets that may be of interest to us. And those gene sets, it's a priori classification of genes into biologically relevant groups or sets. And those sets may be members of the same biochemical pathway, genes annotated with the same molecular function, transcripts expressed in the same cellular compartment. Your gene set may be defined as co-regulated or co-expressed genes, or, for example, genes are located on the same cytogenetic band. So gene set is a set of genes that share particular function. Those gene sets, they are organized in certain annotation databases and ontologies. So annotation database contain sets of genes that share particular functions. And it's a structured prior knowledge about genes. Obviously, if you have your genes that have been identified as differentially expressed, you may go to PubMed and type each gene individually and learn something about each individual gene, but it will take quite a long time and quite a lot of efforts from you to understand the function of every gene and make some conclusion about what those genes that have been identified as differentially expressed, what are they doing? So for that purpose, we have annotation databases and ontologies, and gene ontology is probably the most popular database that organizes information about gene function in a nice hierarchical structure. So that's what I have on the slide. An ontology is a formal hierarchical representation of concepts and relationships between them, and we will see how it looks like. The objective of gene ontology is to provide controlled vocabularies of terms for description of gene products. And those terms are used as attributes of gene products, facilitating understanding some functions about them. You can query gene ontology database and get functions of those genes and make some conclusions about them. This is how gene ontology hierarchy is organized. So first of all, you can see those three large domains, and I will talk more about them: cellular component, biological process, and molecular function. And down the hierarchy, you have more and more refined understanding about some particular function. For example, biological process pigmentation is a biological process pigmentation during development, it's like a child of pigmentation, and so forth. Regulation of pigmentation during development, and so forth. So as you go down in hierarchy, you get more and more precise functional annotations. I already mentioned that gene ontology has three large domains of, think about those three large domains like three separate hierarchies. You have one hierarchy for cellular component, another for biological process, and another is for molecular function. And here they are listed in the order of how precise they are. So as the name suggests, molecular function will tell you some information about molecular function of the gene. And generally it's like an example may be transcription factor. The gene is a transcription factor, or the gene is a DNA helicase, or the gene is participating in certain signaling pathway. Biological process, it's a more broad characterization of gene function, so it gives you some broad understanding of what gene is doing from the biological perspective. For example, gene may be involved in mitosis or urine metabolism, and generally I recommend using this biological process gene ontology because it's one of the most interpretable piece of information that you will get. Cellular component is characterization of where gene is expressed. So you may for example, you may have set of genes that are all expressed in the plasmic reticulum or in the nucleus, or may be located on telomere regions. So it's just as the name suggests, where your genes are located. So it may be informative, it may not be informative. Again, generally, when you analyze functions of your genes, you will be most interested in biological processes. That's a website for gene ontology, and you can explore gene ontology, how it organizes online. You can do enrichment analysis on the website. You can paste your gene IDs in the text box, select which domain you want to use, and biological process is selected by default. You can select an organism, click submit, and you will get a list of functions that are enriched in your list of genes. So gene ontologies are not created equal. So obviously it would be nice if we can have infinite amount of money and we can do lots of experiments and understand functions of each and every gene. But it's not the case, and very often functions of genes, they can be inferred from electronic annotations, like it's the last part here. The function of a gene may be inferred from electronic annotation, or from curator statement, or from author statement. The best information about the function we can get from experiment. But it's not always the case. Most of the ontologies, most of functional annotations, we can get from computational analysis. So that's this cartoon overviews where your functions are, where your ontologies are coming from. And this figure shows you orange color, the proportion of non-experimental annotation, and blue bars show the proportion of experimental annotation for different organisms. And even you can see that even for Homo sapiens, more than 50% of annotations, they are not non-experimental. They are derived from some computational analysis, from data mining, and that's pretty much the case for all other model organisms, for mouse, for rat, for chicken, zebrafish, and so forth. Even for C. elegans. You might think C. elegans is very easy to culture, very easy to make experiments and understand which genes correspond to which functions. It's not actually the case, and it might be like 80% of the genes. They are functions of those genes. They are derived from some computational analysis. Why do you care? Because when you interpret your genes, you want to use those ontologies that have been experimentally validated. So that's what you need to pay attention when you perform enrichment analysis. This link I put there. So again, I mentioned that you should pay attention which ontology you use. And this website with link over here allows you to download, for example, ontologies with experimental evidence only. These are like experimental evidence codes. EIP, I think it's inferred from electronic, I forgot what is it, but basically what you are interested in, experimental evidence only and direct functions. You are not interested in inferred functions like from computational analysis. You can of course use them, but again, it's not as precise as to use experimental evidence only for your functional enrichment analysis. There are many different ontology databases for different organisms, for mouse, for rat, for Drosophila, C. elegans, zebrafish, and so forth. All of those databases provides you with knowledge which genes doing what in each and every organism. You have also ontologies for plants, for yeast, and so forth. So it's just if you work with some other organisms than human, you can use those databases.
Is there generally, and this is my ignorance, like one data for the mouse genome, or are there competing databases put out by different sources?
They are not really competing, but they are sort of complementary, because mouse genes, they are very similar to humans, and very often like functions of mouse genes, and if the mouse genes is homologous to human gene, You can say, okay, in mouse, this is a tumor suppressor, so it must be tumor suppressor in human as well.
But are there multiple like mouse genome databases?
But they are multiple databases. It's simply for convenience. So because mouse genes have different nomenclature than human genes, that's pretty much why we need so many databases. The same for rat, the same for Drosophila. It's even more involved. Arabidopsis has their own gene nomenclature. So, but they are talking with each other in terms of sharing functions of homologous genes. So, another database which provides some information about gene sets besides gene ontology. So, MCDB is from Broad Institute, and they provide sets of genes that correspond to they are structured in those like sections. I think I have those sections right here. So hallmark gene sets. They are those gene sets are manually curated from literature. For example, some high-profile paper that has been published, and they defined those set of genes correspond to stemness. For example, like stemness signature. And MCDB people, they look for this paper, they extract gene set corresponding to stemness, and they put it as a hallmark gene set. So it's a very high quality collection of gene sets and very interpretable ones. So that's what you may want to use for interpretation. I jump to C6 and C7 sections because it's similar to hallmark gene sets, except that C6 is oncogenic signatures. Same principle. People look at cancer-related publications. They find gene signatures corresponding to some cancer-related or tumor suppressor processes and put it as a one list is like list of tumor suppressors, another list of oncogenes, and so forth. And C7, immunologic signatures. We can study autoimmune diseases, and for those immunologic signatures, you can have signatures of T cells, B cells, different subtypes of those cells, and also they have their own gene signatures, which are assembled in this particular section. You can also have gene ontologies. Gene ontology, I believe, let's see. This is C5 is gene ontology. So gene ontologies, remember, the gene ontologies, each function contain like gene set corresponding to this function. So those gene sets corresponding to different gene ontologies, they are assembled in MCDB as well. It has computational gene sets. Motif gene sets is also interesting because you may have a hypothesis that you have a set of genes that may be regulated by one particular transcription factor or several transcription factors. So you can use those gene sets from this particular set, from this C3 section, whatever we call it, and identify whether your genes are regulated, share are co-regulated by one particular transcription factor, or you can get several particular transcription factors. So that these sets of genes are designed for that purpose. Positional gene sets, we already talked a little bit about it. It's where your genes are located. You may, for example, have a hypothesis that you study some cancer and you have a hypothesis that the whole arm or particular part of your chromosome is gone, and you want to identify whether genes that are located in this region that has been deleted, are they downregulated? That's what you use positional gene sets to answer this particular question. So MCDB, basically, it's a very useful database with nearly all gene signatures that you may use, including gene ontology, including oncogenic, immunologic, and other hallmark gene sets. Pathways. Pathways is another set of databases that contain gene sets that participate in one or several signaling pathways, signaling or metabolic pathways. So this is a formal definition of what is a pathway. An ordered series of molecular events that leads to the creation of new molecular products or change in cellular state or process. Genes often participate in multiple pathways. It's like I already talked that one gene has multiple functions. So one gene can also participate in multiple pathways. So pathway databases, they are similar to gene ontology databases. And probably the most popular is KEGG pathway database. KEGG stands for Kyoto Encyclopedia of Genes and Genomes. It's a very nice collection of biological information about pathways compiled from published material. It's a curated database, manually curated database, and the main advantage of it, it provides nice interpretation of your data. Basically, you get lists of pathways that your genes may be participating in, and you can easily understand what those pathways are all about. KEGG is not free. In 2012, they stopped. They started to provide their services on commercial basis, but still there are databases that contain information from KEGG, and I will talk about them. Again, KEGG provides you with nice interpretation of your data. This is how KEGG pathway looks like. It's like, it doesn't look very fancy. It looks like a set of genes that are connected together from up to down signaling propagation. But still, you can see how each gene interact with each other and may influence with each other. KEGG allows you to overlay your genes on this pathway. For example, if you have a list of differentially expressed genes, you may overlay them on this pathway and see where your differentially expressed genes are located. And obviously, for example, if p53 will be highlighted as red, you may think that it's pretty interesting because this gene is a very important one, and you can see if it's differentially expressed, it may affect several upstream and downstream processes. Besides KEGG, there are other databases. Reactome is another well-known database. It's mostly about metabolic pathways. It also has signaling and other biological processes, but Reactome, as the name suggests, is about chemical reactions. It's mostly about metabolism. This is how Reactome website looks like, and you can browse pathways like drill down from cell cycle, cell cycle checkpoints, DNA damage checkpoint, and so forth. And you can go down to some specific signaling parts of your path. There are definitely more, like with gene ontologies, like with MCDB, which contains multiple sections. There are other databases for pathways, pathway commons, and it has more than 42,000 pathways. PathGuide, Wiki Pathway, BioCarta, another very popular one, Consensus PathDB. I would probably not mention that very much, but those that are listed, they are in order of popularity and may also be used for enrichment analysis. If you work with enrichment analysis, most likely you use some programming language, which most frequently people use is R. And in R, those databases that I have been talking about, they are available. For example, MCDF, it's the data frame containing these molecular signature databases in a nice format that you can use for enrichment analysis. Annotables allows you to convert different gene IDs from one format to another. And PathView allows you to visualize your genes over a pathway. And this is how visualization looks like. So you can get, this is a KEGG pathway, and you can see like which genes are differentially expressed and how they overlaid on this pathway. And you can see like those genes are differentially expressed. So nearly every channel has genes that is differentially expressed. So you can make nice figures, and this may help you to understand where your differentially expressed genes are located. Obviously, if you have something like ingenuity pathway analysis, ingenuity is much easier to use and it provides much nicer pictures. It's commercial though, and if you don't have ingenuity access, so pathway in R is a good option to visualize your data. Genes to networks. Networks is another type of enrichment analysis you can do. I will talk a little bit more about it. Here I just mentioned some databases, and with links, it provides you with information. Genes are not linked by functions in this case, but genes are linked into networks based on different properties. Co-expression, protein-protein interaction, and those databases have this information that allows you to build gene networks. Again, I will talk a little bit more about it. So far I have been talking about databases that define functions of genes. Now, how do we use those databases to perform enrichment analysis and to understand something about our differentially expressed genes? So there are three types of enrichment analysis. They are split like first and second and third generation. First generation is traditional overrepresentation analysis. I will talk a little bit more about it. They are hypergeometric distribution-based tests. Let's come back to it. Second generation, I will actually talk about it, so I'll skip those definitions. Just third generation, that's what I just mentioned. It's a network or topology-based tests. So you are looking at the network and you are looking at the position of differentially expressed genes within this network. So let's talk about first and second generations enrichment analysis. First about first generation enrichment analysis. This is actually the first self-contained test, and competitive test is the second generation. So what is self-contained enrichment analysis? Self-contained enrichment analysis is you are looking at gene set and gene set only. So think about you have hypoxia pathway, and you have 100 genes in hypoxia pathway, and all you care in this self-contained tests. I have 100 genes for hypoxia. How many genes among those hundreds I identified as differentially expressed? And for example, if out of 100 you identify 50 genes that are differentially expressed, you may calculate p-value. Is it significant or not? 50 genes, you may say, well, it's kind of half and half, it may not be significant. But if out of 100 genes for hypoxia pathway, 90 genes changed their expression, you may say, you can calculate p-value, that it may be pretty significant enrichment. So this is what this self-contained test is all about. Competitive test is this part of the figure, the right part. And what we care here, we care about our gene set, and we care about the rest of the genes. So when you look at your gene set, again, think about hypoxia pathway with 100 genes. And you identify 50 genes that are differentially expressed. So you really don't know whether it's significant or not. But you can look at the rest of the genes and identify what proportion of genes in the rest, what proportion of genes in the rest of the genes also are annotated as being from hypoxia pathway. So you look at the proportion of genes that are differentially expressed and in hypoxia pathway, but also differentially expressed, but not in hypoxia pathway. So you look at the proportions, and we look how it looks in statistical terms just in a second. So you calculate significance p-value. I already mentioned hypergeometric test, and a little bit how hypergeometric test works. Think about it in very simple terms. You have m genes. M is the total number of genes. For human, it will be 20 to 25,000. That's your universal genes. Out of these universal genes, you have, let's talk about N. N is the number of differentially expressed genes. This is your N. You identify your differentially expressed genes. You also have J genes. It's the number of genes in the functional category. Think about it. Those are our hypoxia pathway genes. And K genes is the number of overlap. How many genes that are differentially expressed and in hypoxia pathway. That's all you need to perform hypergeometric test. How hypergeometric test works? You can calculate the expected value. What's the proportion of genes out of your differentially expressed ones that will be in hypoxia pathway? That's the expected value. But if in reality you observe a value that is larger than your expected value, this functional category or pathway said to be enriched. And this is the ratio of enrichment you can calculate, and for this ratio of enrichment, you can calculate significance p-value. So again, back to this question. You can calculate what's the number of genes you would expect by chance given those settings, given those numbers. But then you can look what you detect in reality, given your number of differentially expressed genes and given the number of real overlaps. And if it's larger, then you can calculate p-value for it. And this p-value is calculated using this 2x2 contingency table. You may have seen it in connection with Fisher's exact test. And Fisher's exact test and 2x2 Fisher's exact test is equivalent to 2x2 hypergeometric test. It's the same 2x2 contingency value table, and calculations are different, but p-value still will be the same. What you build 2x2 contingency table, this is 2 columns and 2 rows. This is just the total and the total. So you build your table, the number of differentially expressed genes and in gene set. This is this K. Then you calculate the number of genes that are differentially expressed but not in a gene set. For not differentially expressed genes, you calculate how many are in your hypoxia pathway or any other genes. And for not differentially expressed, you calculate the number of those that are not in gene set. Having this 2x2 contingency table, you can calculate p-value of your enrichment analysis using hypergeometric distribution. And I won't go too much in this formula. It's a sum over its construction with all those binomial coefficients. It's a hypergeometric probability mass function for hypergeometric distribution. If you read about it a little bit more on Wikipedia, it has a nice explanation. But basically, if you have those numbers for your experiment, which are very easy to get, you can easily plug them into this formula and calculate the significance p-value. It answers the question, probability of having K or more genes from the category in the selected N genes. You can also calculate under-representation. Why we care about under-representation? Think about tumor suppressor pathway. And you did some intervention, some drug treatment. You would expect that in tumor suppressor pathway, some genes will be changed. But it may happen that none of the genes, or very, very few genes that are changed in tumor suppressor pathway. Then you can hypothesize that something is going on. You put your drug, but it didn't affect tumor suppressor pathway. So it must be that you get some functional response that you need to investigate further. P-value calculated very similarly, except in the previous slide, remember K is the number of overlap and N is the total number of differentially expressed genes. That's the sum that goes from K to N, but in case of under-representation, the sum goes from 0 to K. Again, I won't go into it for the sake of time. You calculate this significance p-value given this 2x2 contingency table, and if you work in R, you use this function Fisher.test. It's Fisher's exact test. more precisely would be to use this function, hyper-G-test from geostats package. It performs conditional hypergeometric test to account for a hierarchy of gene ontologies. Remember, I showed you gene ontologies are organized as a hierarchy, and if you do just enrichment analysis on each function in this ontology, you can get a lot of significance functions, but they are kind of redundant. Remember, like pigmentation during development, they are similar things. So this hyper-G test accounts for this hierarchy and gives you more precise p-values. How those functions work, I put a link here. You can see the actual R code. You provide it with gene lists, and it will give you p-values. So that's pretty much what I'm using, and it works pretty efficiently. So hypergeometric tests has its problems. Remember, I told you that genes share multiple functions. Genes also share multiple pathways. And this leads to problems in Fisher's exact test calculations. Basically, what you have, genes are not independent. So, and this not independent wording violates key assumptions of the Fisher's exact test. So, pathways overlap, and this may throw off your p-values, may make your results look significant, but they actually may not be significant. There are ways to account for it, and those ways are pretty much how you construct this 2x2 contingency table. I put a link here for the paper that describes it in more details, but it's again, the point here, you can correct for pathway overlap by properly constructing your 2x2 contingency table. What I describe is like a simplistic approach that works and is relevant, but you can do better. And there are software packages that can do it. We will mention them as well. Second generation, gene set enrichment analysis. It goes by name GCEA, or it has different flavors. I will, at the end of the lecture, I will list one package that has all those different, implement all those different methods, but gene set enrichment analysis is like an umbrella term for the second generation type of enrichment analysis. What's so special about it? What's so special about GCEA? GCEA doesn't look for just differentially expressed genes. It looks how much they are differentially expressed. So you are considering that this gene is highly differentially expressed. This gene has a little bit smaller fold change. So you consider the rank of your genes that are differentially expressed. And this rank is important. Obviously, if you have genes that are highly differentially expressed, you can think about it. You need to put some more weight on those genes, and because they are more important, they change their expression a lot, and you pay more attention to them. How GCEA works? It's a very elegant idea and very simple idea. You sort genes by log fold change. So you organize them from very highly differentially expressed to very low differentially expressed. And actually on the other spectrum. So if you organize them by fold change, on the top you will get genes that are upregulated. At the bottom you will get genes that are highly downregulated. So it's a ranked list of genes by fold change. And now what you do, you think about it. Let me see. If I look at the picture, if you think about this gene list like a ranked list of genes, on the left you have the most upregulated genes. On the right you have the most downregulated genes. And what you do, you walk down this list. Don't have it here. Calculate running sum. Increment when gene is in a set. Decrement when not. This is pretty much what it says. Think about it again. Hypoxia pathway. If we have our gene list that is ranked. You take the first gene and ask a question. Is this gene in hypoxia pathway? Yes. And you calculate, okay, checkbox. This gene is in hypoxia pathway. You go down the list. The second gene. Is it in hypoxia pathway? Yes. You get another checkbox. And you go down the list and you collect the sum of those checks. How many genes, if you walk down to the list, are in hypoxia pathway? Intuitively, if you have genes that are highly differentially expressed and in hypoxia pathway, they all will be concentrated on top of your gene, of your ranked gene list. And you can calculate, this is lower right portion of the figure shows you how this enrichment score, ES, is calculated. You go down the gene list and you calculate the number of checkboxes. For example, this gene is in hypoxia pathway, another in hypoxia, another. But eventually they won't be in hypoxia pathway, and you decrease this enrichment score, and you can calculate the maximum of this enrichment score. This will be enrichment score of your ranked gene list in particular pathway. And you can do it for multiple gene lists from a pathway or from gene ontology, and calculate significance p-values. That's what this is what I just explained you by words. It's pretty much written on the slide, how you do this. Also how you calculate enrichment score that reflects the degree to which set S is overrepresented at the extremes, top or bottom of the entire ranked gene list. Again, you have your ranked gene list, and you look which pathways are enriched in genes that are on top or at the bottom. Remember, at the bottom, you have genes that are significantly downregulated. And downregulation may also be important. It's just the opposite of upregulation. And those downregulated genes may also be enriched in particular pathway, which you may be interested in. The actual p-value is calculated with Kolmogorov-Smirnov statistics. I won't go much into detail. The best place for it, I think I have a reference for it because this is a landmark paper. It's very nicely explained. This is just a formula to calculate enrichment score, and again, just what I said. If you find genes that are in gene set, you increment this enrichment score. If you find genes that are not in a gene set, you decrement this gene score, and what you get maximum of those scores. And for this maximum score, you calculate p-value. You can calculate p-value by permutations as well. So basically for those enrichment scores, you calculate p-value and put significance of enrichment for particular gene set. That's about the same thing. It's pretty much repeating the same, just in another figure. You have your ranked gene list. You walk down through this list. You identify maximum enrichment score, analyze significance, and I have not talked much about it. I should mention multiple hypothesis testing. Remember that when you test your gene sets for enrichment, you are testing whether your gene set is enriched in one pathway, another. You are doing those multiple tests at the same time. And just by random chance, all those sets that you are testing, some of them may be just found significance by chance. So the list of p-values that you are getting out of enrichment analysis, that your gene set is enriched in one or another pathway, you need to correct this list of p-values by multiple testing. The most frequently used is false discovery rate. It's a matter of the whole other seminar, but basically take home message: when you do enrichment analysis, do multiple testing correction. There are other methods to perform functional enrichment analysis, which includes linear model-based methods like camera, correlation-adjusted mean-rank set test. I will just mention them because they are modification of classical enrichment analysis in a linear model setting, camera and ROST. I mention them simply because they are easily available in R, and they are kind of more recent development of enrichment analysis. So, briefly about third generation enrichment analysis. And they are network or topology-based analysis. And what is it? Think about impact analysis. Impact analysis incorporates topology of the pathways, and think about it like that. You have pathway that starts from some receptor that transmits the signal downstream and so forth. If this receptor, if the gene encoding for this receptor is downregulated, you might think, oh, it's very important because if this receptor is down, downstream signaling won't work at all. So that's significant. But if you find that this receptor is fully functional, the gene for this receptor is fully functional, but some genes are differentially expressed, you really don't know whether this signaling is shut down or not. So this is where you consider the topology of a pathway. And there are tools that have been developed for it, Pathway Express and SPIA. SPIA, I think, is still a viable approach. That's at least implemented in current methodologies. So these are tools to perform gene set enrichment analysis. I mentioned GCN is a good way to perform enrichment analysis, and you can download a Java application on your computer, plug in your ranked gene list, and perform enrichment analysis and get nice interpretable results. G-Profiler, it's online. You just plug your gene list and get some results. TopGene and Metascape. Metascape allows you to perform enrichment analysis of multiple gene sets. Think about multiple experimental conditions. You can get one list of differentially expressed genes, another, and another. And you want to know which processes are enriched in one, but not in another. You can put them in Excel spreadsheet, put in Metascape, and it will give you an answer. David, it's probably the oldest gene set enrichment analysis. It's, I think it's from 2012. Or no, it's even early. In 2012, they stopped updating it, but recently they probably got some funding and updated it, and now it's fully functional and fully updated, so it provides quite interesting results. I'll skip Fry because it's not so important. These are R packages to perform gene set enrichment analysis. I already mentioned geostats. Limma, it's a general purpose package that allows you to perform normalization, filtering, differential expression detection, but it also includes functions for performing function enrichment analysis. Some of them I already mentioned, camera, ROST, and there are some more options. So you have many choices to perform this enrichment analysis. I personally would use Cluster Profiler for functional enrichment analysis, but it's again, for the sake of completeness, I give all those options. The most recent development in R packages, Enrichment Browser R package. It allows you to analyze microarray and RNA-seq data. You can also perform normalization, differential analysis, and then you can perform all those types of enrichment analysis that we have been talking about. The first, second, and the third generation of enrichment analysis, overrepresentation, enrichment, and network analysis. So functional enrichment analysis, the very first, it's the first and second generation. So you can see GCA, you can see camera, you can see ROST. It has all those different options in one package, and you just change the parameter and see how your results change. It can also have perform network-based enrichment analysis. Many different options, including SPIA, including all other options, and it has nice visualization options. So this is the latest development in pathway enrichment analysis. I think they published a package just this summer, sometimes in August. But I looked at it, it works very well and provides you with nice way of testing those, nice way of interpreting your data. So I have some references that are not many, but all of them are important in terms of understanding how you interpret your data. This blog from Dave Tan, if you go to this to his blog and search for gene ontology enrichment analysis, you will get lots of practical advice how to perform enrichment analysis in R. And there are some review, a couple of review papers, which again the goal is to provide you with information how to perform enrichment analysis from practical to statistical points of view. So on this, I have, that's pretty much all I wanted to say. Let's see if we have some questions, if somebody can type questions online. We don't have many people in the room, but let's see if we have some questions. Obviously, people in the room have questions. So.
For the GC analysis with the ranking of the genes, does it have to be by fold change, or can you rank them like any way you want?
You can rank them. It's a very good question. The question is about when we perform gene set enrichment analysis, like this. How do we rank genes? We generally rank by fold change. By fold change, you can get up-regulated genes on top, down-regulated genes on the bottom. Is it the only way? Can you rank genes by some other means? Yes. For example, you say, well, I don't believe that my genes are just acting in one direction, that all up-regulated genes must be in a pathway, or all down-regulated genes must be in a pathway. It may be a mixture of them. And you can rank genes by p-value. Irrespective of fold change, the gene may be up or down regulated, but it may be significant. And you can rank them from most to least significant. And the question in this case will be those genes that are highly significant. Doesn't matter whether they are up or down regulated. The matter is they are highly significant. Are they enriched in any particular pathway? So this will be another way of performing this enrichment analysis.
Thank you.
Can we also do like a sequence alignment based enrichment? For example, if you have an LNC RNA that binds to a mRNA, so can you see like the sequence based alignment for that?
Sequence-based ranking, I'm not sure whether, because basic, well, you can, but it depends on the type of experiments you're doing. When I'm talking about it, I'm sort of focused more on differential expression analysis. But if you do some sequence similarity analysis, you may rank your genes like they are most similar by sequences, or they bind to particular sequence, or they regulated by particular microRNA, and you can rank them by anything you want. Again, it's just a matter of what kind of metrics you choose to rank those genes. But then enrichment analysis will do the rest in terms of saying, out of your ranked gene list, is it something special about it? So, anyone should have this presentation as a PDF on Google Drive, and obviously you have my email address as the first slide. Any questions you may have, just email. And again, I'm always happy to answer practical questions in terms of enrichment analysis. And it's again, the whole point is to get better understanding about your gene lists.
Thank
So I
you.
guess we stop here.
Yep. Thank you.